Alright, beginning of chapter 3, Cellular Level. Remember quiz 1 and 2, or quiz 1 is going to be over chapters 1 and 2, so just make sure you look over your study goals and be able to answer those questions. Don't forget, email me with any questions. So with cell theory, it is stating that basically cells are the building blocks of all plant and animals, and that basically all cells come from the division of pre-existing cells. So they have to come from a prior cell. Cells can't just come out of thin air. There are the smallest units that perform all vital physiological functions. There are over trillions of cells. Each individual cell will maintain homeostasis at the cellular level. So overall, basically, the whole organism maintains homeostasis. It's the coordinated action of many, many cells. So when we look at cells, remember that they're the functional unit of the body. Given the research techniques that we have now, we can see these physiological activities happen, transport of substances from outside of the cell to in, through a variety of mechanisms. And it's usually looked through a microscope. But another thing that's important to remember is that cells are not just perfect little circles. They're different shapes, sizes, even some neurons of our nervous cells can be seen without the microscope. They're very long, and they have different sizes to them. And just remember that they're just not perfect little circles. With this, with the digestive tract, they are square or rectangular. The heart is going to have the heart muscle, and the skeletal muscle is going to have striations. So those are all really important in our cellular function. And then the sperm has got this little tail that's coming off the end of it called a flagella. So just be mindful that they are in variable sizes, different locations of the body, and so really we kind of go back to structure dictates function, and the function will help dictate the function, or the structure. So if you are a cytologist, basically you are going to study the structure and function of cells. There are two main classes: sex cells, or germinative reproductive cells, and this consists of the male sperm or the female oocyte, the cell that develops to an egg. And basically everything else is going to be a somatic cell. So soma means body, and it's all of the cells, eyelash to your toenails, except for the sex cells. So figure 3.1 is going to be a really good picture of like the overall organelles and basic structure of a typical cell. Not every cell is going to have each of these structures, and some cells are going to have more structures than others, and certain cells may not even have these centrosomes or centrioles in particular. We'll talk about more of that in just a little bit. But be able to refer back to this image on figure point 3.1 so you can kind of get a layout of the cell. So when we look at the typical cell, it's surrounded by extracellular fluid, which is basically the interstitial fluid of the tissue. It's the watery medium that surrounds the cell, and the outer boundary is going to be the plasma membrane, or the cell membrane. Those two terms are interchangeable, and that separates the cytoplasm from the extracellular fluid. This helps perform vital functions, but when we talk about the cytosol, be mindful that the cytoplasm will contain the fluid cytosol in the intracellular structures collectively known as organelles. Now, there are three key differences between the cytoplasm and the extracellular fluid. Cytosol will have an increased number of suspended proteins. Remember, inside the cell, we are doing lots and lots of enzymatic reactions. There are going to be a higher number of potassium ions, and then the concentration of sodium ions are going to be much lower. The cytosol is also going to contain small quantities of carbohydrates, which provide energy, and small reserves of amino acids that make proteins, and lipids, which can be used as energy when carbohydrates are not available. So when you think about the cytoplasm or the cytosol, it has a lot of functions where the extracellular fluid is basically just a transport medium. There's no reserves stored in the extracellular fluid. So this is what a plasma membrane looks like. You can see that it's a phospholipid bilayer. With this, I need something to point with. Give me 2 seconds. This will be good enough. So the phospholipid bilayer right here, you see the phosphate heads that are hydrophilic. They love the water. You'll see them on opposing sides with the hydrophobic tails meeting inside each other. Inside you'll also find cholesterol to help give the plasma membrane more structure and support. You'll find proteins, different types of channels, and different types of proteins to help identify the cells to others. You'll find carbohydrates, these glycoproteins, inside the cell you'll find the cytoplasm with the cytosol, and then this part is going to be the extracellular fluid. So when we talk about the cell membrane or the plasma membrane, it's a phospholipid bilayer. It's a semi-permeable membrane that allows some things in but keeps some things out, and you'll find proteins, lipids, and carbohydrates there. So when we look at this, remember that the plasma membrane is not rigid or uniform, and again it has the phospholipid, the phosphate heads going to the outside, more hydrophilic, where the tails are meeting inward towards each other, bilayer, and those are the hydrophobic tails. So the extracellular fluid is on the outside and the cytosol is on the inside. So metabolic waste can go out, but the nutrients stay in by selective transport. So functions of the plasma membrane are really important. There's a couple of different types. So basically this phospholipid bilayer is extremely thin, but it does provide barrier. It helps keep enzyme and structural proteins inside the cell. Number 2, it's going to be the bodyguard of the cell. It's going to allow ions and nutrients enter, but able to have the ability to eliminate waste and release secretions. Remember how I said that the plasma membrane is a semi-permeable structure, which means that certain materials can cross the membrane to the inside, but the reverse is true as well. So the waste product of that cell can leave in order to prevent a buildup of toxic waste. Now, the extracellular fluid is going to be a affected by pH, so there's going to be chemical signals that will be very sensitive to the outside environment. And then lastly, the plasma membrane is going to anchor the cells and tissues. For example, skin cells at the outermost surfaces are going to be very tightly attached to one another, but others at the deepest level are going to be very tightly attached to extracellular proteins. The phospholipid bilayer is 2 layers with the phosphate hydrophilic heads towards the watery environment, both sides, with the hydrophobic fatty acid tails inside the membrane. This is going to be a barrier to ions and water-soluble compounds. So again, it's a lipid layer. So water-soluble compounds need a way to enter in some other way. We need to talk about the different organelles. There are 2 broad categories. So the non-membranous organelles are not completely enclosed by a membrane, and basically all or some of the components are going to be at direct contact with the cytosol. This includes the cytoskeleton, the microvilli, the centrioles, cilia, ribosomes, and proteasomes. The membranous organelles are going to be covered by the plasma membrane. It's going to be isolated from the cytosol, and this includes the endoplasmic reticulum, the Golgi apparatus, lysosomes, peroxisomes, and the mitochondria. Be able to identify these membranous and non-membranous organelles and each of their functions. I will ask questions on this on the test. Fair warning. So the cytoskeleton is going to be our cell skeleton of internal proteins to basically be a framework for flexibility and strength. Realize that when you hear skeleton, our cells are not made of bones, but they do provide a lot of structure, a metabolic organization where these key enzyme reactions are going to occur and where specific proteins are going to be synthesized. So all of these are going to be protein tubes and provide a great deal of function. And these include the microfilaments, the intermediate filaments, microtubules, thick filaments, and the microvilli will help increase surface area on the periphery of the cell. So, microvilli, you can see these small finger-like projections come up from the periphery of the cells. We have the intermediate filaments, kind of like right in here, these little purple ones. You're going to have microtubules. You're going to have thin and thick filaments throughout the cells. In muscle cells, you're only going to find the thick filaments. So microfilaments are going to be the smallest of the cytoskeletons. It's basically the thin filaments composed of a protein called actin. It has 3 major functions. Provides additional mechanical strength. It does interact with proteins for consistency. So if we have more microfilaments, it's going to be more of a gelatinous. And then if they're kind of widely dispersed, it's going to be a more fluid concentration in the cell. And then lastly, they're going to pair with thick filaments of myosin for muscle movement. Intermediate is the mid-size between the micro and the thick filaments. So it's the most durable of all the cytoskeletal elements. And that's because it has collagen in it. So, for example, keratin of skin, it allows skin to be very strong but resist stretching. So it's going to stabilize the position of the other organelles, but also provide lots of strength. Microtubules are going to be large hollow tubes of the tubulin protein, and they extend outward into the periphery of the cell from the region near the nucleus. They're going to attach to the centrosome. They're also going to strengthen the cell and anchor organelles to provide more rigidity. They can change the cell shape. They really are important in mitotic division. Form the centrioles, the spindle apparatus, cilia, and flagella. So very, very strong, very movement related. Thick filaments are only found in muscle cells, and they're made of the large myosin protein. And they interact with actin, the thin filaments, to produce powerful muscle contractions. Microvilli are the small finger-like projections of the plasma membrane on an exposed surface. They greatly increase surface area to the extracellular fluid, so you'll really find them in areas of the digestive tract to facilitate absorption. The centrioles in the centrosome are important in cell division. They help direct the movement of chromosomes, and the centrioles will form the spindle apparatus and organize the cytoskeleton. They look like very cylindrical structures composed of short microtubules. Cilia are found where movement is necessary. They are small, hair-like extensions. I think people get them confused with microvilli a lot, where cilia is made for movement and the microvilli are made for absorption. So these small finger-like projections or small hair-like extensions will move fluids across the cell surface by beating rhythmically to move fluids across the cell surface. You're going to find these in cells in the respiratory and reproductive tract. So in the respiratory tract, they're going to move kind of sticky mucus around, trap dust particles. If you have a family member that's a smoker, they could have damaged cilia and they will not be able to move fluid. And so they'll typically wake up with that morning smoker's cough to kind of clear all of your junk and all the mucus, and that's not being able to be cleared by the cilia. You also see this in the reproductive tract to help move oocytes along the uterine tubes, or from the sperm from the testes to the reproductive tracts. So you're going to find them in various places. And this is what a cilia looks like. So small hair-like extensions. Now, cilia is going to be these extensions that we talked about, but the proteosomes are these little small cylinders of enzymes, and proteosomes are protein enzymes. So they break down and recycle damaged or abnormal intracellular proteins. They help clean them up. Ribosomes are going to be responsible for protein synthesis using information found within the DNA of the nucleus. Remember, we need proteins all the time. We need that repair and other tasks. The number of ribosomes in each cell can vary, and it's for its demand for protein. So we have two different types, fixed that are found on the endoplasmic reticulum or free that are scattered through the cytoplasm. They are composed of a large and small subunit which have ribosomal RNA that joins with mRNA or messenger RNA to create protein synthesis. And we're going to talk about this in detail in just a little bit. And we talked about the proteasomes that basically help disassemble damaged proteins for recycling. Now we're going to move on to the membranous organelles, and that's going to include your endoplasmic reticulum, the Golgi apparatus, peroxisomes, mitochondria, and then the nucleus, with also lysosomes. There they are. We'll talk about each of these in just a little bit. Oh, there we go. So the first one is endoplasmic reticulum. I think if you break this word down, it can sound very foreign if you've never heard this word before, but if you break it down, endo means within, plasm means the cytoplasm, so it's within the cytoplasm, and it's a network of these intracellular membranes involved with the synthesis, storage, transportation, and detoxification. They form the storage chamber within membranes, and there are two types, smooth and rough. Smooth is involved with lipid and carbohydrate synthesis, and the rough endoplasmic, covered with the fixed ribosomes, are active in protein synthesis, and they are going to really be able to enclose these products to transport them out of the cell and to repair other cells. So it's very important that we have functioning endoplasmic reticulum. So here you can see a picture of them kind of very close to the nucleus, because again, we need that DNA out of the nucleus to help create the template for proteins. So you need to be really close to them so that you have this information. The Golgi apparatus is going to be, again, in close proximity to the endoplasmic reticulum, and they are going to form secretory vesicles that are discharged by exocytosis. So with that, you're going to see three major functions. They're going to modify and package those secretions. They're going to create, which will renew or modify the plasma membrane, or they'll package special enzymes called lysosomes for use in the cytoplasm. So I think Golgi apparatus kind of look like dinner plates that are just kind of stacked up, and those membranous discs are going to be called cisternae, those flattened chambers. So, okay, this is a really complex picture, guys, and I really want to kind of go over this a little bit more simplistically. So I'm going to cover it up. And we're going to talk about each piece. Okay, so we have the nucleus that has the DNA on the inside, and you have the rough endoplasmic reticulum, and then the smooth endoplasmic reticulum. Remember that the rough endoplasmic reticulum is going to kind of look like very sandpaper in appearance, and it's going to synthesize and modify proteins. And it's going to package these proteins to the Golgi apparatus. So in the rough endoplasmic reticulum, you're going to see this protein that's made using the information found from the DNA, and it's going to transport it through a transport vesicle to the Golgi apparatus. So the smooth endoplasmic reticulum, you're going to find these making lipids or carbohydrates, You're really going to find them really in great amounts in the liver and the kidney, because the smooth endoplasmic reticulum is really important in detoxing or inactivating drugs. And you'll find them a lot in muscle cells and neurons to help repair those things. And they're just very important. So, if you had to think about the endoplasmic reticulum as anything of a city, if you had to think about like the cell as the city, and each organelle has its role within the city and how it functions and how it works, the rough endoplasmic reticulum and the smooth is going to be your factory. So when you take these transport cells and you move them over to the Golgi apparatus, again, the Golgi apparatus is going to modify and package these proteins so that we can kind of change them into something that we can use. So we can make them more complex, we can make them renewed or modified. So we'll kind of, as they kind of come in through this receiving side, they're going to go out to the packaging side and be able to be sent out in a secretory vesicle. So it's a little bit changed, but not much different. And if you had to compare the Golgi apparatus to the city, it's going to be the post office or the Amazon. It's going to modify or package these products. So they're not going to make anything there. They're just going to send it out. So these secretory vesicles are going to be secreted out of the cell. And you can see that the protein's coming out. So the protein's going to be expelled out of the cell by exocytosis. The lipid can be expelled out to repair other cells, other cell membranes. And one thing that we haven't talked about is the lysosome. And again, we'll kind of breach on that in just a little bit. All right, so endoplasmic reticulum is going to be the factory with transport vesicles going to the Golgi apparatus with the secretory vesicles. And then finally, it's going to be expelled out of the cell through exocytosis. Okay, so now we can kind of look at this more complex picture and maybe hopefully understand it a little bit better. Okay. Okay, so protein synthesis begins when a gene on the DNA produces messenger RNA, the template for protein synthesis. So you can see that the DNA will open up and send this message out. All right. The messenger RNA can leave the nucleus and attach to basically a free ribosome in the cytoplasm or a fixed ribosome in the rough ER. So that message can go out and go on a free one or into the rough endoplasmic reticulum. All right, cool. Proteins are constructed on these ribosomes, and basically then they can release them out into the cell. Okay, cool. All right, not really sure how that's doing. All right, when a protein is synthesized on a fixed ribosome on the endoplasmic reticulum, it's threaded through these hollow tubes or the cisternae, where it will begin to get more complex and get into more of a 3D shape. So then basically the endoplasmic reticulum is going to do this through those transport vesicles containing the modified protein. The transport vesicles will kind of now move into the Golgi apparatus, where now it's going to modify and package this new product for use. So as it goes through the Golgi apparatus, it's going to further modify and package it into something called the secretory vesicle. So basically it's going to go from the receiving side to the shipping side. And this is done through the secretory vesicles creating a new protein, lipid, or on the different side can kind of become a lysosome. And that's going to contain digestive enzymes. You're also going to have the secretory vesicles that will fuse with the membrane and empty their products out through exocytosis. And then basically the lipid ones or the carbohydrate ones are going to kind of renew the plasma membrane of itself or others. So we kind of talked about endoplasmic or the lysosomes being filled with these powerful digestive enzymes. They're kind of small spherical bodies. And in the city, we are talking about them being the disposal surface. So they have several functions, but overall, they're going to be responsible for providing an isolated environment that will help prevent against potentially dangerous chemical reactions. So if they see viruses, bacteria, damaged organelles that are no longer functional for the cell, they're going to break them down and eject them out through exocytosis. Basically, they're little internal stomachs of our cells. Peroxisomes are smaller than lysosomes, and they carry a different kind of enzymes that will neutralize toxins. They can break down fatty acids, free radicals, and they're really, really important. They replicate through division. So the flow of the cell is going to be from the endoplasmic reticulum through the transport vesicles to the Golgi apparatus, through secretory vesicles, out the cell membrane through exocytosis. And this is going to allow change, remove or recycle segments of the plasma membrane. It's a continuous communication by the vesicles. And again, in an actively secreting cell, you may find that the entire plasma membrane could be changed in an hour. So it's very adaptable and changeable. The kidney-shaped bean, the mitochondria, I'm sure you've heard the powerhouse of the cell. It's that organelle responsible for energy production. So the number of mitochondria can vary with a particular cell's demands for energy. So like muscle cells have a great deal of mitochondria because they need that energy to move. Red blood cells really don't require a lot of energy, so they don't have any mitochondria. They have a smooth outer appearance, and they do have a fold in the inner membrane called cristae, and that is where energy will take place in creating it. So why do we need ATP? Well, cells need this energy to break these high energy bonds under very controlled conditions. So it's a way of making more energy. It's taking the chemical energy from food to produce the energy molecule of ATP. So here you can see that it's very kidney-shaped bean, and you have these inner folds that have like a little membrane itself. Now, this is going to be a kind of a complex slide. Chapter, I think it's 26 in semester 2. Basically, there is an entire chapter on this one slide here. So we're just going to go over the basics of it and understand that mitochondria creates energy production, and most of it will occur inside the cell. So the first steps are going to be glycolysis, and that is done in the cytoplasm. So basically you're taking sugar or glucose into pyruvic acid, and you only make a very small amount of energy. This pyruvic acid will enter in this citric cycle, but depending on your professor or who's teaching the chemistry class, it may be called the Krebs cycle or the tricarboxylic acid or the TCA cycle. And this will happen inside the matrix. So the pyruvic acid will enter this citric acid cycle, and it will take the byproducts of CO2 and kind of kick them out. And we'll use some of this into the electric transport chain. So you'll take all of these extra hydrogen molecules, remember those ions, and they'll enter this chain where it will just make a ton of ATP. And it will create a byproduct of water, but it requires oxygen to come in for that byproduct of water. So it's called aerobic metabolism. It's called cellular respiration. It needs oxygen to really work. So the mitochondria will use the oxygen to break down food and produce ATP, but the end product will be ATP, but you'll have byproducts of carbon dioxide that you can breathe out, and water, which our cell will always use. So again, this is going to be on your lab sheet. Look at this slide. It is very complex, but just to recap, glycolysis will happen outside in the cytoplasm. You'll take glucose from food, turn it into pyruvate. The pyruvate will enter into the mitochondria and enter the citric acid cycle. So you're going to kick out CO2 as a byproduct, and you're going to use the hydrogen to enter into the electron transport chain, and you're going to get this oxygen that comes in. Another byproduct is going to be H2O, and you will hopefully get a ton of ATP. Now, one of the most important organelles is going to be the cell nucleus. It's going to be the largest one. It's going to contain DNA and enzymes essential for controlling cellular activities. It's the control center. The nucleus has the information needed to make over 100,000 different proteins. Cells without a nucleus will disintegrate within 3 to 4 months. So you need cells to survive with a nucleus. It's surrounded by a nuclear envelope. It's a double membrane, and it has these nuclear pores that communicate to the cytoplasm so they can send the message out. So inside the nucleus, you'll contain nucleotides, enzymes, chromatin, surrounded by this double membrane with multiple pores. It controls metabolism, storage, and process of genetic information, and the control of protein synthesis. DNA contains all of the information to build and run organisms. The nucleoplasm is basically the fluid containing ions, enzymes, nucleotides, and some RNA. It has a supportive nuclear matrix, one or more nucleoli, and chromosomes. So basically these chromosomes will help kind of provide that genetic information. The nucleoli are related to protein production and made of RNA and synthesize ribosomal RNA with these subunits. And then the matrix supports it. So chromosomes are DNA bound to histones, so they're tightly coiled. These are found in dividing cells. Chromatin is loosely coiled DNA in cells that are not currently dividing, and that's pretty typical, so they're kind of more of a relaxed look. The supercoils only happen in mitosis, giving chromosomes their characteristic X shape. So, DNA is going to have instructions for every protein in the body. They're a very unique sequence of amino acids. The gene will contain all of the triplets or DNA instructions needed to code for a specific polypeptide. So a single DNA molecule will be pairing DNA strands held by those hydrogen bonds. Remember that double helix. And they have the complementary nitrogenous bases: adenine, guanine, cytosine, thymine, and that's that chemical language. This triplet code will be 3 bases to 1 amino acids. So basically, it will specify the identity of a single amino acids, but it has to be read in groups of 3, and they're called codons or triplets. Remember that a gene is a portion of that DNA strand that functions as our hereditary units. So they're going to give you information for specific proteins. It's a very complex thing, but we're just going to hopefully go over the basics of it. The nucleus will contain the chromosomes that contain the DNA, and DNA stores that information for proteins, and that proteins will determine the cell structure and function. So when we look at this, gene activation is uncoiling the DNA to use it, and there's going to be basically a temporary remove of those histones. It's going to begin the temporary disruption of weak hydrogen bonds between the two, so it's going to allow the 2 DNA strands to help separate, and it begins with a promoter that starts the message and a terminator that ends the message. When we talk about transcription, I want you to think about what that sounds like, to copy or rewrite. So when we talk about transcription, we're writing down the message, the synthesis of RNA from that DNA template. So we're taking the messenger RNA from the DNA in the nucleus that cannot leave it, but messenger RNA can. And this will happen through the messenger RNA, and it will carry the instructions from the nucleus to the cytoplasm. The formation of a protein is done by translation, and this will help translate the message into something that we can understand, into the language of proteins. So when we talk about transcription, it's writing down just from the DNA. It's done in the nucleus by RNA polymerase, the enzyme, and you create a strand of messenger RNA. When we talk about translation, it's basically translating the messenger RNA into understandable terms to create a protein. Protein is that functional polypeptide constructed using the messenger RNA codons, the messenger of those 3 units. And the sequence of these codons or triplets are going to determine the sequence of amino acids. And these complementary base pairings of anticodons, so the message that we translate or write down, will provide the amino acids in sequence. And so the whole process will have the rough endoplasmic reticulum and the Golgi apparatus producing the protein and packaging the protein. So remember, key things with gene activation and protein synthesis is DNA cannot leave the nucleus. Transcription ensures that the mRNA is basically the same. Oh gosh, sorry. Oh gosh. And you know what? That'll probably be a good end to this, and we'll talk about this a little bit more in the next lecture. I hope you found out that I figured out how to combine the 2 Chapter 3 videos, so into one. Again, please email me, do the lab handout. We'll resume Lab 3 next week. But make sure you fill out that handout to turn in next week. All right, and I will look forward to seeing y'all Thursday for the lecture and quiz 1. All right, thanks.